Macromolecules. In this video, you're going to learn how the structure of macromolecules is related to their interactions in carrying out the cellular process. In other words, how does the structure provide support or evidence of its actual function? And specifically, carbohydrates, proteins, lipids, and nucleic acids. One protein specifically we'll take a look at are enzymes. Macromolecules are these very large organic molecules. Organic means that they contain carbon and they make up all living things. The four main ones that we will learn in biology are carbohydrates, lipids, proteins, and nucleic acids. We can't live without them. They run our body, they hold information on how to run our body, and they provide us with the energy to do so and the structure to form new things. Everything we will learn for the rest of the semester will relate to macromolecules. They're the foundation of understanding how biological things work. Structurally, most macromolecules are what we call polymers that are made out of monomers. Polymers are the larger structure, and monomer is the individual component. If we were to look at monomers and polymers more specifics, you would see that a monomer is the small kind of basic unit, almost like a brick. And a polymer is the larger structure that is composed of all of those monomers, kind of like a brick wall. So a monomer is to a brick as a polymer is to a brick wall. And if you use that analogy, a nucleic acid is to a small component that makes up DNA structure. Here is my nucleic acid, and it is the smallest component that functions, that physically makes up my strand of DNA. So how do these molecules form? Well, polymers, were made from monomers. Most of the time it's a dehydration reaction. It builds a polymer by linking together monomers. It does this by removing a water molecule, hence dehydration. So an example of this is when your body has more glucose than it needs. So it combines excess glucose molecules to what we call stored glycogen. You have one monomer, which is this guy right here, and another monomer, which is this guy, and we're going to remove some compounds attached to it, this particular one right here, OH, and an H of this monitor, which when you put those together is water. And when we remove that, so by removing the water, the two monomers are linked together by this leftover oxygen. If this dehydration reaction happens again and again, then monomers will just attach to each other and attach to each other and attach to each other, forming larger and larger polymers through a dehydration reaction. Now, how we break those polymers apart is through hydrolysis. We break big polymers into smaller monomers. We do this by adding the water back. By adding the water back, you actually break the bonds. This happens, for example, in your digestive system, as enzyme breaks down those complex starches into more simple sugars. That's why you produce a lot of saliva when you're eating. Your saliva contains those digestive enzymes, and they're the first part of digestion as they're starting to already break down these large complex starch molecules into simple sugars so they can be easily digested when they hit your stomach.